right, so we are now transitioning to Chapter 7, reminding you that we're not skipping Chapter 6, we're just moving it to the start of Unit 3. It's going to be all things nervous system in the second unit. Chapter 7 and 8, in my opinion, really be concisely kind of put together into a single experience, but it's broken apart, so it won't take us too terribly long to get through Chapter 7 nor Chapter 8. So we'll wrap those up, if not today, on Tuesday, and we get into specifics about the central nervous system. When we look at Chapter 7, we're going to look at basic nerve cell properties and electrical signaling, which you had an introduction to lab last week. Right? So it was a bit of a crash course, and I mentioned we're going to review it, and that's what we're doing here in Chapters 7 and 8. We're going to review the major structural components of the nervous system, and then talk about how information is functionally transmitted directionally-wise between the central and the peripheral nervous system. We're going to review the structure of a nerve cell, and then we're going to add to the function of those. And so these first two items, we'll probably spend the bulk share of today actually talking about these first two chapter objectives. We're going to review as well different types of ion channels and where they're likely to be found in a nerve cell, reiterating handout number 4. We're going to reintroduce the structure and function of myelin. We should get to that objective today, and then review the resting membrane potential, how it's established, the role of the pump, and the leaky channels. What will be new to us is item 6. Incorporating a new term called a graded potential, and we're going to contrast that to an action potential. We're going to see how to differentiate those, and that will be reiterated in handout 5, which you just received. And then we'll bring in the role of the gated channels, their contribution to an action potential. And then the rest of Chapter 7, we're going to see how an action potential can be conducted along a myelinated or non-myelinated nerve. And then we're going to identify the role of the refractory period. So we may not get maybe these last few items covered today. We may have to transition over to Tuesday to finish it up. What I want to do here is use this image from your textbook to kind of give you a physiology relationship between all the cell structures in our nervous system. And so this should be figure 7.1 in your textbook. And if you look real close, you'll see some color differences. You kind of see the more tan versus the more beige area. This is representing the different functional nervous systems that we have. And so if we want to functionally divide our nervous system, we have what we'll refer to as the central nervous system. Which organs comprise the central nervous system? The spinal cord and the brain and the spinal cord comprise your central nervous system. What is our other functional division of our nervous system? We have the central and we have the peripheral nervous system. We said the central nervous system is made of the brain and the spinal cord. What is the peripheral nervous system made of? Cranial nerves and spinal nerves. Your spinal nerves and your cranial nerves comprise your peripheral nervous system. Those are the organs of the peripheral nervous system. Whether you're evaluating a cranial or a spinal nerve, you can really subdivide these nerves into their function. And our cranial or spinal nerves are functionally either afferent or efferent. Or I'll say afferent, so we really are enunciating. We have afferent cranial nerves, we have afferent spinal nerves. What does that mean? Okay, so the directionality of the nerve signals are going towards the central nervous system, the brain or the spinal cord. So what kind of information could be conveyed to the brain or spinal cord afferently? Sensory information. Very good. So if I talk about an afferent nerve, I'm talking about a sensory nerve, whether it be from the a cranial nerve or a spinal nerve. If it's afferent, it's sensory, conveying sensory information from the periphery of your body to your brain or spinal cord. If we evaluate it a little more, we see that there's some subdivisions of sensations that our afferent nerves could tell us about. What kind of sensations could our afferent nerves tell us about?
Heat.
Okay, so like heat. What else?
Touch.
Touch. We can sense heat, we can sense touch, we can sense temperature would be heat.
Oh.
And other, not just heat, but cold temperature too, right? So very good. What are you saying? No, you were probably right. I was just speaking over you. Taste, very good. We can sense taste, temperature, either hot or cold. What else? Can we sense? Smell, touch,
And
sound.
is it balance?
Balance is, you can sense your balance or your, yes.
Equilibrium.
Yeah, so equilibrium, balance to it. Did you say taste? Yes. Sight, right? You're using your vision. You can also sense what's going on inside of your body. You may not cognate it though, right? So your body is monitoring how your heart works, right? And so on and so forth. And so we can subdivide our senses into three subcategories. The first that you guys listed, like your touch and your temperature and etc., fall under a category called somatic afferent senses. Somatic, so like the surface of your body. What could you feel at the surface of your body? Heat, touch, pain, physical, cold, right? Whatever. You could sense these things, right? At the somatic. The somatic afferent gives us body surface types of sensations, including not only just like your skin, but your skeletal muscles. You feel a stretch in your muscle. You're using which types of nerves? Somatic afferent, if you're sensing it. What about things like sight, taste, hearing? Olfaction would be a sense of a smell. How can we categorize all of those together? Those would be called your, well, they're a type of special senses. You ever heard that word? Special senses? Your special senses include your vision, taste, hearing, sense of balance. What am I missing? Smell.
Says vision, hearing, equilibrium, smell, and taste.
Very good. Thank you. So all those are modified. They're special senses. They're specialized in our body. So we can not only monitor the surface of our body somatically, but we can sense special types of stimuli, light, sound, and etc. A third type of sense we don't always think of maybe immediately, but it's called your visceral afferent nervous system. Where would this monitor?
Heart.
An example would be, say, like the heart, your internal organs, so your smooth muscles, your digestive system, and etc. So your internal visceral organs have to be monitored in order for your brain to know what's going on down there in the bowels and whatever. So let's tie into what we studied in lab last week. If I want to detect, if I have nerves that can detect somatic sensations, what do those somatic afferent nerves need? Receptors. If I want to detect light in my special senses, I need light receptors. So all of these nerves would have specialized receptors on them. But they're nerves, and so in the presence of an adequate stimulus, these things could depolarize to their Did I hear it? They can depolarize away from the rest and reach their threshold to generate an action potential. And we now have an idea after chapter 7 that our nerves, when reaching an action potential, can release what? What can a nerve cell release when it's excited with an action potential?
Sodium.
Neurotransmitter. Okay, nerve cells release neurotransmitter. So our sensory afferent nerves receive stimulus because they have specific receptors. If they depolarize the threshold, they can release neurotransmitter. Where does it look like the neurotransmitter would be released?
Axon terminal.
From the axon terminals where? Do you think your afferent nerves need to terminate? What are they trying to communicate with? The brain or the spinal cord. So they probably ought to terminate in the
Spinal cord.
spinal cord or brain. Just depends on which nerve you're looking at. All right, afferent nerves can release neurotransmitters somewhere in our brain or our spinal cord. So if we release a chemical messenger at a cell in our brain or spinal cord, that cell in your brain or spinal cord better have a receptor, which would hopefully bring this cell up to, allow it to depolarize up to its threshold, generate an action potential, and maybe even release its own neurotransmitter. They're still just nerve cells. The brain and the spinal cord, if we go back to the relationship of maintaining homeostasis where a receptor, control center, and effector, if we look globally at that pathway, the brain and the spinal cord will act like your control center. The sensory nerves would be acting as the receptor, and the efferent nerves will control the I don't use that word. If I have afferents that sense in, I have efferent that would control what?
Motor.
Some type of, it could be motor, some type of command like an effector organ. It could be a muscle, may not be. Let's put this in perspective, hopefully, with this example. Let's say you see this light. You're detecting the presence of the light in this room. You would use your photoreceptors and your optic nerve. Optic nerve will depolarize to threshold, generate an action potential, release neurotransmitter in your brain. Cells in your brain will receive the neurotransmitter. Those cells will,
Decide what to do.
before they can even decide what they do, they would depolarize to threshold, generate an action potential. And there's going to be other cells along the way, but eventually we're going to get to an efferent nerve. And let's say this light, you're like me, and you get a friend, the light is blinding you. Okay, so what do you think you need to do? Squint, or even just the pupils by themselves would first just kind of constrict. So my brain would activate specific efferent nerves. The light goes in, detected by my optic nerve. Optic nerve releases neurotransmitter to my brain. My brain selects a nerve called the oculomotor nerve, which is a type of efferent nerve. It will depolarize to its threshold, generate an action potential, release neurotransmitter to what could respond? Does the nerve do the eye movement? The muscles in your eye. So the efferent nerve would release neurotransmitter on your effector organs. In this case, it is a muscle or set of muscles that allow for the pupil to constrict and reduce that light input. I hope that wasn't too much of a divergence, but I just want to kind of put it in perspective of the relationship of these different nerves. So if there's something that my body can sense, I have a specialized receptor at the end of an specialized afferent nerve. It can talk, if you will, to the central nervous system by releasing neurotransmitter. The nervous system, the central nervous system, is going to act as our control center. And if a change is necessary, it's going to have to activate a completely different set of nerves that we can just simply call what? I want to do something, I need to bring into action some efferent nerve.
So it's kind of like the effector, like can it check it on the same way?
You can think of them around the same thing. I don't want you to think of them as the same thing because the efferent nerve has to talk to the effector. So the effector would be the muscle, would be a gland, or whatever the case may be. But it is the link between the central nervous system and the effector. So if you want to kind of think, well, they're related because they are. The efferent nerve talks to, if you will, if you let me use the word talks to the effector by releasing neurotransmitter.
So like the message in the entities like with efferent nerves are moving through the central nervous system to the peripheral nerves.
Correct. But she said, so the way that, if I make sure I'm not summarizing what she said, in order to kind of talk to the effector organ, the only way the central nervous can do this is by way of the efferent nervous system. Is that kind of what you said?
Yeah, and just like getting like the direction, right? Like it's always gonna be going from like central nervous to the peripheral nervous.
Yes. Yeah, so will the direction efferently always be central nervous system to peripheral? Yes.
Okay.
If you're looking efferently. So if you want an outward command, you need to use the efferent nervous system. And the arrows in this image are really good. They do represent that directionality of talking or communication. It's not that simple though. You see all these subdivisions, right? The efferent nervous system is subdivided because we have so much variation of way that we can control our body. The one that you're most familiar with, whether you've thought about it or not, is called the somatic efferent nervous system. Notice the name sounds the same. Somatic means what? Body, but this is the efferent nervous system that controls the body. I'll come back to that and describe that. So our efferent nervous system operates either somatically or what?
Visceral.
You can step yours back. Either somatic or autonomic are the two major subcategories. Somatic, we said, means body. So what about your body could you control? This voluntary control of your, clarify that you can't voluntarily control all of your reflexes, like salivation, for example, or tummy growling, right? What?
Of your muscles?
Which ones?
Your
Skeletal muscles. So the somatic efferent nervous system gives us control, a command to control our skeletal muscles, which we could voluntarily control. So you would have before today probably would have called it the voluntary nervous system. The proper term would be the somatic efferent nervous system. So if I want to move across this room back and forth, I'm using the somatic nervous system, which controls, I need to clarify this, not the skeletal system, but the skeletal muscles. Be sure to clarify that. It's your skeletal muscles that you can somatically, efferently regulate. So if I can somatically or voluntarily control my skeletal muscles, that must tell me there's parts of my body that autonomic that I would control involuntarily. And so voluntarily somatic, involuntarily autonomic. We'll caution you from saying automatic. They're easy to mix up, but autonomic means self-regulating. The involuntary nervous system. If you look at this one, it's even more diverged, okay? The autonomic, the involuntary nervous system itself is also subdivided into two precise categories. They are called the sympathetic and the parasympathetic nervous systems. The sympathetic and the parasympathetic nervous systems. These two are terms which you may not have ever used before, but you've talked about them before if you've ever talked about somebody's response to something. Sympathetic, if you've ever talked about the fight or flight response, if you've ever said that, you are talking about the sympathetic nervous system, a subdivision of the autonomic efferent peripheral nervous system. So when you're in scenarios when you're scared or nervous or exercising or whatever, your sympathetic nervous system is prevailing. Conversely, should we have our body in this phase of excitation all the time, like sympathetic exercise? No, we shouldn't. Most of the time, we should be under which type of control? Parasympathetic control. And I'll refer to as your rest and digest nervous system. So the question is then, why do we have these two subdivisions of the autonomic nervous system? Why do we have two subdivisions of this autonomic involuntary nervous system? So we can't consciously control it, so it kind of just happens under the regulation of the brain and spinal cord. What else?
Kind of bring you up and for contract that, like if we need it,
Yeah, yeah. No, it was fine. She said they kind of bring you up or bring you down or they contract when you need it. And so the autonomic nervous system controls parts of your body that you can't consciously control, like your heart, your endocrine glands, and so on and so forth, or the digestive activity of your body. Someone asked about this dashed line. Since I increased the font on here, so your cardiac, smooth muscle, and your glands can be controlled by each of these subdivisions. This gives us precise control of your heart, your smooth muscle, your glands, using only the autonomic nervous system. And so like a balance of one another. So I could speed my heart up when I'm exercising, but I could also slow it down when I'm not exercising. And so we're going to elaborate the relationship in chapter 11. We'll call dual innervation. A lot of our organs are dually innervated. Another subset of the parasympathetic nervous system, right, regulates the GI tract, basically. But we won't talk about it so much. We'll talk about it more, I think, when you go into like your nutrition class, looking at the physiology of all the internal organs, digestive system. Unfortunately, we don't have to think about if I'm anxious during a test, I don't have to worry about knowing the answer, and then also telling my stomach to stop rumbling and whatever, right? All those things. So I spent a bit of time on here because I think it gives you a nice survey of the entire Unit 3 of where we're going. And so if you ever feel lost, come here and put yourself in perspective. Chapter 7 and 8 talks about any nerve, wherever it may be, about its structure and difference of a graded potential versus an action potential, etc. Chapter 9 will talk exclusively about the central nervous system. Chapter 10 addresses the sensory input, and then chapter 11, the efferent motor control of the rest of the body. So it's a nice little roadmap of where we're going. What questions do you have about this arrangement before we review nerve cell structure?
Why are there empty boxes?
Where? Oh, just in and out. I think your book has in and out.
Oh.
I was just trying to cover them up. Sensory afferent conducts in and efferent conduct out. Sorry. Didn't even catch it. What I'll do is spend some time looking at the nerve cell structure. This image is straight from your textbook, repeated from handout number 4. And I noticed something was missing going through the presentation yesterday. So at the very end of your table, if you could add this last item, we're going to talk about the role of voltage-gated calcium channels. So when you got to add the voltage-gated calcium channels in the end. So before we get into any detail, I want you to answer this question. Do all nerve cells look like this?
No.
Not at all. But this is just a generic looking nerve cell. It's a little bit more specialized looking than the round cells or the square cells that we've seen up till now. We're talking about any cell, right? Now we're saying, well, these are nerve cells that have specialized structures. But they are cells, so they have a membrane, they have receptors, they have channels, they have pumps, they have a nucleus with DNA, they can transcribe, they can translate, they can make lipids, right? They can, right, so on and so forth, okay? So don't forget all of Unit 1 application here to this specific type of cell. What I want to do is just look at the basic anatomy. We'll just go through this list in order and look at not only the anatomy, but then the function or the physiology of these things. And so most of our cells, many of our nerve cells will have processes called dendrites. Where are the dendrites here?
Cell body.
They branch off of the cell body, where left or right? The leftmost side. And it's these things. Those are just membrane extensions. Those are individual dendrites. They're processes or extensions. What are some functions of the dendrites? They could receive, for example, neurotransmitter from other nerves. What else could they receive? What else could our nerve cells receive?
Ions or gas.
So ions, so voltage. They could receive chemicals like neurotransmitter. Voltage is a result of ions. What else could they receive? Chemical stimulus, electrical stimulus,
Physical.
physical or mechanical stimulus, or thermal. So, sound familiar? The dendrites receive different modes of stimuli because this is where we have a high concentration of our sensory receptors. So you're going to have your chemically gated channels, your voltage gated channels, your thermal gated channels, your mechanically gated channels, etc. are poised here at the dendrites because that's their job, is to detect stimuli. And so that's where all those receptors are going to be localized. They branch off of a region of the cell called the cell body. I call it the cell body. You could just as well, more precisely, call it the soma, S-O-M-A. The soma is the cell body. And what's going on here at the cell body? All the organelles are there, right? So all that general cell structure stuff here inside of the cytoplasm, mitochondria, oxidative phosphorus, you name it. We talked about it for 4 weeks, happening inside of that cell body. But what you may not be aware of is the membrane of the cell body may also have what? What else could be in the membrane of the cell body?
Channels.
Channels. What kind of channels?
Gated.
Gated channels to receive different types of ions, or we could receive different types of. An example would be neurotransmitter. We said that would be a chemical, or ions, which would be a voltage, or
Chemical.
Just stimulus, all of the different.
so what does our cell membrane have? at the cell body, it also has receptors. The stimuli can be also detected at the cell body. In the cell body, the membrane also expresses various receptors. The nerve cell body, opposing generally where the dendrites are, would extend into an area which we'll call the axon. But I want to focus on an area called the hillock, right where it extends out into the axon. It's called the axon hillock. The axon hillock probably also expresses what? It's all repeated so far. It probably receptors. So the axon hillock can also receive various neurostimuli, neurotransmitters, ions, heat, whatever the case. Yeah. All right. So we can also receive stimuli at the hillock. Okay. The hillock, though, is identified uniquely in that not only are those receptors there, but really there's a very high concentration of voltage-gated sodium channels. So we see our first real concentration of voltage-gated sodium channels here at the hillock. What this means to us is that this is the part of the cell that's really sensitive to any change in depolarization. And if we want to simplify that in even more, we can say this is where the nerve cell's threshold is established. This is anatomically where the threshold is established for a given nerve cell. So again, our axon hillock has a very high concentration of the voltage-gated sodium channel. So if some stimulus causes the cell to depolarize enough, the voltage-gated channels at the hillock will open and initiate that all or none principle that we'll review called an axon potential. The hillock will then extend into the axon itself, which is shown here. And what is the job of the axon?
Transfer the action potential.
To transfer or conduct the action potential from the cell body towards the direction of the, which way do we move?
Axon terminal.
To the terminals. So the axon can conduct action potentials because it has what? What does it have to allow for the conduction of a potential change?
Myelin.
Do it.
Myelin.
Not all nerve cells are myelinated, so they may or may not have myelin. Gated channels. What type of gates? Voltage gated. Sodium channels, voltage gated, potassium channels. This axon is capable of conducting action potentials because of the presence of the voltage gated sodium and the voltage-gated potassium channels. Flip open. The axon may diverge, like we see here, the branching, to form various, so it may not be just a simple one axon, one terminal. It may have multiple axon terminals on any given nerve cell. What happens at the axon terminal? What's the axon terminal?
Receives the action potential and releases the neurotransmitters.
Okay, so you listed two things: receives the action potential, releases neurotransmitter. So let me clarify. It can also conduct an action potential, so it also has what?
Receptors.
Let's change the word. What kind? voltage gated sodium channels, voltage gated potassium channels. There's something new here too. Next, last in line, the added thing, there's also what here? The voltage gated calcium channels are localized to the axon terminals. So that the axon terminal is going to release its neurotransmitter.
So it can receive an action for the potential and conduct.
I would use the word conduct. So it's just a continuation. It's a continuation.
And he said something else, what did he say?
Uh,
About the axon terminal.
the storage and release of neurotransmitter. An action potential is going to be continuous. It starts generally at the hillock area and then continues down the axon all the way to the terminal. So it's not like a new action potential at the axon. It's one that's going to continue down the length of the cell. So the axon terminal is what releases our neurotransmitter. Just kind of a flashback to this Tuesday's discussion, alluding a little bit to the calcium. Remember, is that calcium can act as a messenger? In this case, calcium messages the nerve cell to release its neurotransmitter. Some cells have myelin, but not all. Is this cell myelinated?
No.
It is not. If you want to myelinate it, you can. But what is it? If it's there, what does it allow for the cell? More efficiently, faster, right? Conduction. I'm just going to take it at face value today, or right now, and then hopefully we'll get to there today and see what's going on. So if you want to myelinate your nerve, you can put, I'm just going to draw some cell. You wouldn't have a rectangular cell, but it's cells that grow and wrap around it. So they insulate little segments of your nerve cell membranes, so that the surface of the membrane is all covered up. Or I should say sections of the membrane are all covered up. But we have little exposures to it. And so the benefit is to having myelin is just faster conduction of the action potential. You can get from the hillock to the terminal faster with myelin than without. We've introduced some of these. We've talked about voltage-gated sodium, potassium, and calcium channels. They open in response to changes in voltage. And we've translated a little bit, hopefully in our brain, we know that voltage change really comes from a change in ion concentration. So if we change ion concentration, we change the voltage, and we could open or close specific voltage-gated channels. Voltage-gated sodium channels, when opened, will let sodium flow which way? Into the cell. And if sodium flows into a cell, the cell would do what? Depolarize. Very good. So when a voltage-gated sodium channel opens, sodium flows in, causing depolarization. Where would you find these?
The axon hillock.
Hillock and the body, the axon, terminal, and even the dendrite, so throughout the cell. What about voltage-gated potassium channels? When the voltage changes and these things open, potassium would flow out of the cell The polarity change is causing the cell to, so you would use the word repolarize, or maybe even hyperpolarize. Very good. See, all that confusing time in lab paid off, didn't it? I hope so. So an open voltage gate of potassium would let potassium out, and the cell is either going to repolarize or hyperpolarizing, depending on if it's at rest or not. I'm going to hop down to the voltage gated, where would we find our potassium channels? Throughout the cell as well. I'm going to hop down to our voltage gated calcium channels. When the electrical charge changes, these will open and calcium would flow, which way? Into the cell. Yes, they're positively charged. Yes, the voltage is going to change some, but then really we want to focus on what next. Which one? Yes. What does calcium influx cause at a nerve cell?
Like the first stuff you're talking about, with the kinases and everything?
That could happen. That's probably not, well, yes. Let's keep it. And then in the very end, when calcium floods into the axon terminal, we get this pathway going, and then what happens? Exocytosis of the stored neurotransmitter. A pathway is initiated. It's real fast, though. We mobilize those vesicles to release neurotransmitter. So calcium influx at the axon terminal causes exocytosis as a neurotransmitter. We just use sodium, potassium, and calcium using their gradient. So before we can use them, we have to pump them. So we have to establish the gradient. So your cells throughout the membranes would be a sodium, potassium, various sodium, potassium pumps, and calcium pumps throughout, creating those gradients for the ions. Let's just review the sodium, potassium pumps. How many? 3 sodium out to every 2 potassium in. Very good. Establish that gradient so we can use them later. And the calcium pump must pump calcium out. If we let calcium in and cause exocytosis through a gated channel, the pump must pump it in the opposite direction, would be towards the extracellular fluid. We also have throughout the cell membrane, all over, we have leaky potassium and leaky sodium channels. And these directionality-wise are the same as the voltage-gated channels. So leaky channels leak potassium out, and leaky sodium channels leak sodium in. Very
Make that song go away.
good. No, don't make the song go away. Hopefully it'll remember. Pump's pumping, the leaky channels are leaking. To help, are we okay? Or what questions you have over nerve cell structure, function? I had a really good question in class. Someone asked yesterday. They're like, "What about these membrane proteins? Do they actually go through the whole?" I saw someone like with something like piercing. I think of like a javelin stick or something of a protein piercing the entire cell. Do our cells pierce through the entire, or do our proteins pierce through the entire cell? They're just little integral membrane proteins that take up a tiny little segment of a cell membrane in a various sections. So I think you have an idea. I've got your little reminder that the way our nerve cells are going to work is by using electrochemical gradients for primarily sodium and potassium, but certainly we can see calcium coming into play using our leaky channels which are always open. Our gated ion channels, whether they be chemical, voltage, mechanical, or thermal, and then of course the establishment of those gradients, excuse me, by using the sodium-potassium pump. So weekly announcement, we can't brain dump everything that we use, right, learn throughout the semester. So we're putting it into action now. So that was a generic looking cell. If we were to contrast it to some, kind of look a little bit in the structure, we can look at structural classification. And so we'll, looking at figure 7.5, you should see that not all nerve cells look like that awesome looking one that we just saw. There's some modification. Some cells are referred to as bipolar, pseudo-unipolar, or multipolar. The biggest point of confusion which we may have is the term bipolar. That is not in reference to the disease. It's in reference to just the appearance of the cell, meaning it looks like it has two poles, so it's opposite, right? So if you look at it, the dendrites almost look like the axon terminals. The cell body is centrally located in this case. So they have two ends. Conversely, some cells may be referred to as pseudo-unipolar, where the cell body is offset from the axon. You're going to see these when we look at our, been in lab, we talked about the sensory afferent nerves on the dorsal side of the spinal cord, and we talked about the ganglion. If you were in lab, we talked about the ganglion. Those dorsal nerves, your spinal nerves, exhibit this pseudo-unipolar, where the cell body's actually closer towards the terminals. So it's just really just different from what we've seen before. And then what we've kind of run with so far is using an example of the multipolar example of a nerve cell, where we have the dendrites branching off of a central kind of a cell body and then a long single axon that may or may not branch. That's a structural look at our nerves, but we want to study the what? The function. So we're going to simplify it. We are going to call them either one of three things. Our nerves are either what? Going back to our roadmap, either sensory or
Autonomic.
we could even, our nerves are going afferent, efferent, or even in the middle would be afferent to something, efferent from something. What's the something? That would be our control center, right? So our, another word we'll use are interneurons. So functionally speaking, we have afferent nerves. So our, functionally, whether it's pseudo-unipolar, whatever, we have afferent nerves. They convey what type of information? Sensory information. Words. In the middle, we have interneurons. If you go up the prefix, inter means between. Okay, these are the cells between our afferent and our efferent. The afferent nerves convey sensory information. The interneurons act as our what? Control center. They're going to establish a set point for whatever they're there to process and take information in from the afferent nerves, and they may call to action various. Third functional category are efferent nerves. So functionally speaking, I'm gonna say a nerve is either afferent, an interneuron, or an efferent nerve. Whether the efferent nerve is somatic or autonomic, maybe we could just say, well, it's efferent. Maybe if we were taking any specifics away. Does that make a little bit of sense? I do want to turn your attention to figure 7.6 because I like its anatomical arrangement. On your afferent nerves, this may tie into the question you had about the relationship between the efferent nerve and the actual effector organ. You could do the same association with the part of the body that's being monitored. Some part of the body, right, sensory, visceral, special receptors innervate some organ, which is represented by this box. Okay? And then that activates an afferent nerve. But the reason I like this is it illustrates what I try to point out in the roadmap. Our afferent nerves terminates in the central nervous system. That talk to, if you will, the dendrites or cell body of our interneurons, which talk to the cell bodies and dendrites of our efferent nerves. So 7.6 does a nice job of physically showing their relationship as well. Afferent, efferent, I really over enunciate that so that you don't get confused. If you say afferent, efferent, it's easy to kind of get confused by afferent, efferent. So we're okay on the direction and not getting them flip-flop, because you can see they're functionally very much opposite one another. When we also look at the nervous system, not only do we need to look at nerves, we've got to talk about our glial cells. And glial cells are accessory cells in the nervous system, helper cells for the nervous system. They themselves aren't nerves, but they're there to help the nerves of the brain and the spinal cord, peripheral nervous system, whatever, do their job. Our glial cells include astrocytes, oligodendrocytes, microglia, epidermal cells, and Schwann cells. The first four are localized exclusively to your brain and spinal cord. So this first four are central nervous system glial cells. And then that's why I have the asterisk by the Schwann cells. Those must be part of the peripheral nervous system. Accessory cells of the peripheral nervous system. Astrocytes get their name because they look like stars. And again, if you look at a microscope, you're going to they do look processy, but they're not cute little 5-pointed stars. But they look. You see the little extensions from them. These are super important. I was going to say the most important, but it's not nice to give priority of something in the body. Everything's important, so these are really important for the basic metabolic demands of your nerve cells, making sure the extracellular fluid, concentration of, say, sodium, potassium are good in set point range. So they'll go through, and was it this class we said, well, what about neurotransmitter? Could it diffuse out of the cleft and act as an autocrine? So this class had that question? So someone had a question like, well, could a neurotransmitter diffuse out and target itself? I said, well, probably unlikely. And one reason is we have these astrocytes that are kind of like a little bit of a cleanup, a little bit, kind of their job making sure if there's extra neurotransmitter, potassium, whatever, they're gonna help get that out of there. They also help when the brain grows and develops from fetal through early brain growth. They're there to physically just send the nerves on their way, like, all right, you go this way, the nerve cells just kind of grow along. So another important function of the astrocytes is they physically, because remember I said they're cross-branchy like a star, they can reach out and hold, if you will, affix to a nerve axon and affix to a capillary and put them next to each other. Why would it be good that we have a capillary next to a nerve cell? Blood flow, nutrient distribution, gas distribution, and waste. So they will physically orient them together. So astrocytes are really critical for just day-to-day maintenance of your nervous system. Some cells may have oligodendrocytes. These are the cells which form myelin in the central nervous system. So if you look at any white matter in the brain or the spinal cord, it is, right, they have these cells called oligodendrocytes, which wrap around them. A third type of glial cell found in the central nervous system are called microglia. So these are small little cells, and these have immune function. And that they go around with phagocytosis. Remember we talked about phagocytosis That's what they do. They go around and pick up any extra protein bits or whatever your body thinks is potentially toxic. They'll go there and phagocytose it and get rid of it. Another type of glial cell found in the brain and the spinal cord are called ependymal cells. And these cells line the ventricles of your brain. And they help form your cerebral fluid. They help form the fluid. And they're also ciliated. What does that mean? Kind of like the cilia. They're kind of hair-like extensions, right? And so these cells have cilia on one part of them. They're bound in the brain, but then making a tissue. But on one end, they have the apical end, where the fluid is circulating. They have cilia, so they kind of beat in unison to keep your cerebral fluid circulating through it. They not only help form it, but keep it circulating. Those are glial cells of the central nervous system. The only glial cell of the peripheral nervous system are called Schwann cells. And these cells form myelin in your peripheral nerves. And your cranial and your spinal nerves, which are myelinated, have a different type of cells called Schwann cells surrounding them. The Schwann cells form the myelin of the peripheral nervous system. Schwann cells form myelin, and we can kind of look. We've used the word a bit in the last week or so, and visually, you can turn to 7.7b and c in your textbook to compare and contrast the myelin of the central versus the peripheral nervous system. The myelin-forming cells are shown in the purple. And so those are cells that have a nucleus and they metabolize and all that good stuff. But they're specialized to wrap around nerve axons. A key difference between oligodendrocytes and Schwann cells is the ratio of the myelin-forming cell to the axon. If you look at an oligodendrocyte, one cell may wrap around multiple nerve cells. So one oligodendrocyte associated with 1, 2, at least 3 different nerve cells. By contrast, one peripheral nerve may have multiple Schwann cells about it, forming this insulative kind of barrier for.
Is that a person's name?
Do what?
Schwann, a person's name?
Yes, it is a name. Who discovered cells, right? It was involved with the cell theory.
Like the cord, or was it the convoluted?
No, yeah, my history just went out the back door. I think it was, I don't know, my history is, usually I can, we'll look it up after class. So the question is, all right, so we have an idea about myelin. It is formed by either oligodendrocytes or Schwann cells. And these are living cells that associate with nerve cells. Or your nerve cells would be predetermined whether to be or not to be myelinated. So it's not a random kind of thing, or when your body says, oh, I need to speed up, add some or whatever. It's predetermined in your genetic makeup if a cell will or will not be myelinated. And the point is, we put a whole extra bunch of fat, if you will, lipid bilayers around a given cell. So these are some really nice scanning electron microscope images of some myelinated nerve axons. Here we can see at least 1, 2 different nerve cells. The browned out area would be the cytoplasm of the nerve cell. And then what's been colored for us pink and yellow would be the myelin-forming cells. So they actually concentrically wrap around the axon. And then if you look at a nerve organ where you've got bundles of nerve cells grouped together, you can see how well electrically fatty, this nerve organ is and how bundled, wrapped up it is. It's not 100% covered up, and we'll see that